So hello guys and welcome back to Excision Academy. So as part of DNA repair mechanism, this today's topic is about the arms test. Okay. So so what is this arms test? Can anyone tell me why it is being done and it किस तरह से useful है? Tanvi? So this test is actually helping in order to check whether the chemical has got its carcinogenicity or not. Okay. So what does it mean, carcinogenicity or mutagenicity? इसका मतलब क्या हुआ इस शब्द का? Whether that chemical has the potential to cause mutation or not. Okay, so that is what we are going to check. Now, if I say that in the cosmetic industry, also they do arms tests. Okay, so the statement, how can you support to make a judgment or make a supporting statement to this? Can anyone tell me? So we know that in the case of cosmetic, many cell lines as well as animal studies are done. Then only the cosmetic can come into the market. Can you tell me why and how it is being done? Yes, anyone? Akshay, would you like to try? Shruti, you?
Yeah, because since cosmetics involve things that are going to be applied on the skin for long periods of time, they want to see whether it can be absorbed, or if it is absorbed, how much, if it can cause problems, and if so, how much problems, and
Yeah,
so
so
on.
if you see in the case of the cosmetics, the ingredients, those are basically chemicals, okay?
Yes.
And the something which is known as like jo bhi aapka preservative and water will be there. Now, so these cosmetics tests, they also undergo all these tests. And first of all, they also see whether, for example, in the case of white rabbit, okay? So mainly this is first thing they do. White rabbit ke eyes may they inject the cosmetics, okay? Now, I know it looks very brutal, but this is what they do. They inject into the eyes of the white rabbit and then they see how much inflammation or redness is coming. Okay, so this will indicate how much allergic it will be to the human. So the more the redness, more the allergic it will be to the humans. Okay, so these kinds of small animal tests they do before any cosmetic comes into the market. Okay, now same thing. Now, if any chemical is there, you're dealing with any chemical, this kind of test is important because we need to know whether that particular chemical can cause mutations or not. ठीक है, अगर वो हाँ कर सकता है, then it is better to avoid, right? Okay, so let's see. So it is helping in the determining the potential carcinogenic effects of chemicals. Now, in animals, if we do, it is of course going to take so much of time and of course it is expensive. If you see, I'll tell you when I was working at Tata Cancer Hospital, maintaining one male mice and one female mice, okay, in one chamber per day, it was around 560 rupees, okay, per day. अब सोचो कितने दिन उसको रखना पड़ता है. So in a month and also much cost is to come because we have to give them the proper sanitization also, proper feed also, and they cannot eat kuch bhi. So for them also, there are companies who are making feed for them, like proper proportion of carbohydrate, proteins, and fat, etc. Okay, and then they're always being injected with the antibiotics so that they don't get any other disease because they have to be perfectly healthy so that you can carry out your particular experiment which is meant for it. Okay, so that's why it is costly doing in the animals, and of course you want to see how it is going to affect the offspring. So it will take around 3 to 4 months in the case of rat, in the case of mice, to give the offspring. So it is also time consuming. ऐसा नहीं है बैक्टीरिया की तरह 20 minutes में it will just clone or not clone and you can understand the effects of the chemicals. Okay, so it is time consuming and expensive. Okay, however, because most tumor causing agents are mutagens, the potential carcinogenic effects of chemicals can be assessed from their capacity to cause mutation. So this Bruce Arms, who was at University of California at Berkeley, he designed this experiment and he used a bacteria called Salmonella typhimurium. Okay, please remember, ये थोड़ा important है. Okay, Salmonella typhimurium, because this is the host bacteria onto which you are going to check whether the applied chemical agent has got mutagenicity or not. Okay, so ARM says uses a strain of Salmonella typhimurium, which is mutant for the operon responsible for the biosynthesis of the amino acid histidine. Okay, now before I proceed further, what do you think will be the answer? Like, the Salmonella typhimurium is mutant for the operon for the biosynthesis of amino acid synthesis. अगर कोई agent, कोई chemical agent is there, it is having the mutagenicity or carcinogenic effects. So what kind of mutation will it show in this strain so that I'll understand that this chemical agent is mutagenic? ऐसा क्या करेगा? The answer lies in this sentence. क्या होगा इसका answer? What kind of mutation do you expect in the strain if the chemical agent is having mutagenicity?
Its histamine production will be affected.
No. Or socho. So when I say that typhimurium is mutant for the operon responsible for the biosynthesis of the amino acid histidine, इसका मतलब क्या? This mutant cannot. हाँ.
Maybe the DNA is not packed properly. It's the chromosomes.
Akshay, I can't hear you. Your voice is breaking. Yes, once again. Or maybe you can type in the chat. I'll read and then I'll repeat.
Am I audible now, ma'am?
Yes, yes.
So maybe the DNA is not packed properly within the nucleosomes because there won't be any biosynthesis of amino acid histidine.
It's a bacteria. Okay. Bacteria or prokaryotes, they don't have nucleosomes. It is only present in eukaryotes. And it has nothing to do with the packing. When I say Salmonella typhimurium is mutant, इसका मतलब क्या? It cannot biosynthesize histidine. ठीक है? ये sentence clear है? It is a mutant for the operon responsible for the biosynthesis of the amino acid histidine. यानी कि it cannot synthesize histidine. Okay? अगर कोई chemical agent mutant है, तो वो ऐसा क्या करेगा जिससे you will understand that it's a mutagenic in nature to the Salmonella typhimurium?
Maybe it might start producing histidine because the
exactly. Yes, so this is what is happening. So if your mutant is able to synthesize now histidine, it means what? It has caused mutation in the operon. Okay, and now the operon can synthesize amino acid histidine. इसका मतलब क्या हुआ? जो भी आपने add किया, okay, any chemical agent, whatever you have added, has a potential to cause mutation. It means it can be carcinogenic or mutagenic in nature. अभी clear हुआ? अभी?
But ma'am,
हाँ.
You have to get lucky enough that the mutation has to happen over there, right? If the mutation happens somewhere else, then they wouldn't know, even if it is mutagenic.
So that's why we have taken sensitive strain of this typhimurium. अगर हुआ तो it will survive. If it is not, then of course not. Right? The thing is that, see, type. अभी हमने किस लिए bacteria लिया? So that if any mutation is happening, it is going to affect the chromosome of the prokaryotes, right? Introns का सवाल ही नहीं है. So whatever mutations are there, it is going to affect. We'll come to that. Okay, but इतना concept clear है? Is this much is clear? Let's see
Yes,
the
ma'am.
experiment and then we'll come to that. Okay. Okay. So for example, the mutant operon might contain a missense or a frameshift mutation in one of the genes for histidine biosynthesis. So I guess everyone knows what is a missense and a frameshift mutation, right? Okay. So everyone knows what is it, right? Now, the mutant cells will fail to grow and form colonies on solid medium lacking the histidine. So आपको मालूम है क्या होगा if suppose there are colonies and उस medium में अगर histidine नहीं है, so what will happen? They cannot grow. Correct? They cannot grow. But if you add the suspected mutagen and if you see that there are so many colonies, it means what? It has been revert the induced by the mutagen. Okay. So इसका मतलब it has got that potential carcinogenic activity. Okay. Now the thing is that depending on the nature of the mutagen, the reversion of a small number of mutant cells will be either the missense or the frameshift mutation. Okay. Are you getting the point here? जो भी आपका chemical agent है, carcinogenic है, it has the capacity to revert back the mutation. कैसा? Either in the missense or in the frameshift mutation, because of which the later the cells are able to grow. Clear हो रहा है या और confusion आ गया है? See guys, you can talk
Yes,
about
ma'am,
it.
it's
It's
clear.
okay. It's clear, right? So
Yeah.
I hope, see, before you understand this, you should know what is missense mutation and what is frameshift mutation, correct? So who can tell me what is missense mutation? Maybe the other people are not knowing. Missense mutation क्या होता है? Missense mutation, अगर suppose I'm saying it is a missense substitution mutation. Okay, so we know the substitution means mutation ही होता है. तो missense मतलब क्या होगा? अगर कोई ऐसा change, okay, when there is a change of a single base pair or a single nucleotide, okay, because of which it is causing the different amino acid. Okay. Now, for example, if I say U is there. So U, it codes for phenylalanine. Now, if suppose there is a substitution and G it is becoming, then it is coding for something else. Now, I don't know which one. Let us say that it is coding for another valence. Okay. So what will happen? Just because of one particular thing, it has changed. Now, similarly, अगर हम अभी वो operon वाला question देखें, so because of that the operon, maybe operator was not available or the promoter was not available, because of which they were not able to synthesize histidine from the given media. Okay, in the media. Okay, अभी क्या होगा? अगर मैं कोई ऐसा chemical agent use करूं जिसकी वजह से missense substitution हो जाए, okay, so what will happen? The operon will revert back. Revert back मतलब क्या? Already मैंने एक mutant बनाया है, ठीक है? मेरा ये first mutation है जिसकी वजह से operon was not working and that kind of bacterial strain I have selected. अभी second mutation यानी कि second mutagen के वजह से there will be another mutation because of which what will happen? It will go back to its original state. So अब क्या होगा? Double mutation के वजह से it has gone back and now able to synthesize histidine. आया थोड़ा सा आइडिया?
Yes, ma'am.
That's why it is called revert, क्योंकि पहले mutation से मैंने उसको mutant बनाया of biosynthesis of histidine operon. Second mutation यानी कि second mutagen I have added and that was able to go back to its first original state. Okay, so this is how you understand whether it is able to cause mutation or not. अब frameshift mutation क्या होता है? Okay, so these are like छोटा topic which sometimes people think that you guys know and they will skip. Okay, but it's then your duty कि आपको मालूम होना चाहिए कि इस तरह के mutations क्या होते हैं. So frameshift mutation क्या है? Either it is because of the deletion or insertion, the DNA sequence will shift the way it is read, right? So you know that हमारे पास 3 कोडॉन के रहते हैं, right? अगर suppose वहाँ पे insertion या deletion हो गया, so what will happen? The entire reading sequence of 3 codons will change. जिसके वजह से कभी-कभी you have got the truncated or sometimes you have got a long stretch of mRNA or you know the DNA sequence which will be read. तो जिसके वजह से क्या होगा? Either you will have a truncated protein or you will have a good amount of long polypeptide chain, depending on what it is getting inserted or inserted or deleted. Okay, very small topic this one. ऐसा कुछ ज़्यादा है नहीं इसमें, what to say, like to understand much. So there are certain chemicals that cause cancers which are not mutagenic to begin with, but they get converted to mutagens by the liver, which metabolizes foreign substance. So we know that in the liver, now we are talking about the mammals, okay? We are talking about the humans. So we know in the liver, we have got the detoxifying enzymes, okay? Now these detoxifying enzymes, can you name few enzymes which are present in the liver which help in the drug metabolite conversion? Have you heard of cytochrome P450? So these are the cytochrome. So cytochrome enzymes are there. They are one of the detoxifying enzymes. Okay. So कभी भी आप कोई alcohol लेते हो, this is also coming under drugs. Or are you taking anything like anything, any अगर if you are taking abusive drugs जो रहते हैं जैसे कि cocaine हो गया, heroin हो गया, जो हम कहते हैं, right, opiums and all. Okay, so those kinds of drugs as well as the medical drugs if you take. Okay, so all these are toxic to the body. So to get detoxified, it goes into the liver. Okay, now in the liver, these enzymes will convert them into the non-toxic product. But sometimes what happens is that though they are non-toxic at that time, but they might be mutagenic. Getting the point? So,
yes ma'am.
हाँ, so non-toxic मतलब उस time वो cells को affect नहीं करेगा, but since it is mutagenic, it is causing mutations in the DNA and later on the cells are affected. Okay, this is the meaning. Now, what happens is that now whenever a drug, for example, let us say that let's talk about brufen. Okay, brufen हो गया, ibuprofen हो गया, or any other painkiller like paracetamol also they are having this painkiller. Okay, so what happens is that inside the body this goes, okay, and there are certain enzymes like, have you heard of COX-1 and 2? Suna hoga. Or arachidonic acid. Do you guys have heard of these words? So I'll tell you in short. So we have got the substrate as arachidonic acid. Sorry for the spelling. So it is around 20 carbon ka molecule. Now from arachidonic acid, you get the conversion of prostaglandins and you get interleukins. Okay, prostaglandins and interleukins you get. Now whenever there is a pain in the body, किसी भी चोट के वजह से, or you have headache or whatever is it, the prostaglandins levels gets increased. Okay, now we have got the enzymes COX-1 and 2. So यहाँ पे आपके COX-1 and 2 रहते हैं. Okay, now what this brufen or the painkiller does is that they go and target your COX enzymes. Okay, they target this and decrease the level of prostaglandin. इसलिए आपने नोटिस किया होगा कभी भी पेन किलर लेने के बाद आपका हेडेक इमीडिएटली नहीं जाता है. It takes around 10 to 15 minutes and then it shows its effect. Then आपका हेडेक हो या कोई भी बॉडी पेन हो वो चला जाता है, because slowly it is decreasing the volume, decreasing the concentration of the prostaglandins. ये जितना ज्यादा हाई होगा उतना ज्यादा पेन होगा. Okay, ये जितना कम होगा उतना कम पेन होगा, even though the wound is still there or the fever is still there, but the pain will be minimized. Okay, now what I wanted to say is that it is not that directly brufen is going and acting on the enzyme. पहले क्या होगा? First thing is brufen is taken inside the body. Okay, brufen गया, it will enter, it will get absorbed, stomach mein gaya, it gets absorbed into your blood system, right? It goes to the liver, it gets metabolized sometimes, and then जो byproduct होते हैं, even they are go and help in inhibiting the enzyme. Okay, so this is one example. तो अभी हम जब आते हैं drug manufacturing में, so what those people are doing, they make sure that जब brufen है, okay, I'm just saying an example. I don't know right now that brufen gets divided into 2 products or not, that I don't know. I'm just giving an example. So brufen is there when it gets metabolized in the liver. So whatever those products are there, okay, we need to check that also whether it is going to be mutagenic or not. So उससे पहले क्या होगा? In the market, not sorry, in the market, in the drug making procedure, okay, so they have got the brufen, they check for its mutagenic activity. They will also check the metabolites produced after the brufen is degraded into first and second component. So हम इसको भी check करेंगे, ठीक है? Then only when you give all this data results, then only, for example, FDA, Food and Drug Administration of US, will approve the medicine to come into the market. Samjha? You just can't have only the first molecule. You need to see how the enzymes are going to act on this molecule and affect the cells. There's something called pharmacodynamic and pharmacokinetic. आपने सुना होगा in your pharmacology. At least heard about it. Yeah? सुना हुआ लग रहा है?
Yes, ma'am.
Yes. So all this, we also say that as a PK dynamics and all, okay? So why I'm saying all this thing is that because see, just imagine that you just want to join a company after MSc, okay? And there are most of the companies are CRO. What is CRO? And they are clinical research organization, okay? So all over the world, especially in US and Japan and China, also many clinical trials are going on. So they want to make, to make sure that this drug is applicable to all the all the ethnic species. जैसे कि human में तो है ही, okay, human में तो है, but whether it is for Asian, whether it is for American, whether it is for African, उन सब में ये काम करना चाहिए. So clinical trials में क्या होगा? They'll make sure that this particular region of people are also showing the effects. तो CRO का वो काम है. So CRO will come in India. They are in India. They will hire people and they will carry out the clinical trials. ठीक है? And they will make sure, okay, it is also functioning on them. कभी-कभी क्या होता है? The medicine which is working on Americans is not going to work on Indians just because of the changes in the lifestyle we have. So there are chances कि ऐसा हो सकता है. So that's why all this clinical trials are important. Okay. Okay, I gave the broad idea of everything now. I hope ARMS test is clear now. Like why it is important to check the potential carcinogenic effects. Okay. So obviously directly we can't do in the animals, so we go with the cell lines. Okay, in the bacteria में हम सबसे पहले check करते हैं. Then we go slowly when okay, if it is successful, then we go into the cell lines, mammalian cell lines. Then हम उसमें mice में जाते हैं, mice के बाद chimpanzee में जाते हैं, chimpanzee के बाद हम humans में जाते हैं. ठीक है? Okay. So that's all about the ARMS. Let's talk about the repair and the tolerance of the DNA damage. So far, so good, guys. Any questions regarding the ARMS test? Shall we proceed?
Yes, ma'am.
Okay. So now let's talk about the repair and tolerance of DNA damage. So this is what we are going to discuss in the entire DNA repair mechanism. Okay. So first one we have already discussed, that is about the mismatch repair, right? So this is because of the replication error, and we have talked about how this is being repaired in the prokaryotes. Okay, so mute S, L, and H is there. So remember, if there is a mismatches there, so this will be scanned by the which protein? Shruti, S, L, H. इन तीनों में से कौन सा है जो scan करता है पूरे DNA को?
S.
Yes. So S will come and it will scan, and then L and H will come. It will be recruited now with the help of the ATP, which is present on the mute S. Okay, it will activate your mute H, which is a endonuclease. Then it is going to make a cut depending on the directionality, whether it is 5' to 3' exonucleus, or we have got the 3' to 5' exonucleus जो chop off किया जाएगा. Okay, only nick is made by the mutH. Later, these exonucleases will come and remove the extra portion of the nucleotides, okay, the stretch of the DNA. Later, then your polymerase will come and ligase will come and it will reseal the DNA. Okay? In the case of the eukaryotes, it is known as the PMS. Okay. Photoreactivation. This also we have discussed in the earlier lectures where we have talked about the pyrimidine dimers, right? So, wherein which we also have discussed how thymine dimer is formed and it is opened with the help of the DNA photolyase. We have also discussed about the thymine and cytosine dimer, correct? So that's why it is written pyrimidine dimer. So we have got two types. यहाँ पे 5 and 6 carbon will be joined, and here, no, sorry, 6 and 5. Yes, 6 and 5, and here it is 6 and 4. Okay. So I hope you can understand what 6 and 5 and what 6, 4 I'm talking about. If you remember the structure, the cyclobutane ring which is formed, okay? The cyclobutane ring. Okay. Okay. Now, again, it is done with the help of the DNA photolyase. Now, there was this question, okay, it was given scenario that the person has got this pyrimidine dimer formation, okay. Now, the person was always in the dark because he or she thought that it is the skin cancer because of the UV radiation it got. So, the person was always in the dark. Do you think that the person was able to repair its pyrimidine dimer ka problem? Should I repeat the question? Is it clear?
Yeah, they couldn't.
They couldn't. Why? Because
Photoreactivation requires light.
yes. So it means what? थोड़ा और डिटेल में श्रुति. थोड़ा और डिटेल में समझाना होगा तो श्रुति कैसे समझाया जाएगा इस चीज को? Let us say that Tanvi doesn't know. She was not able to attend. So how will you explain this to Tanvi?
So photoreactivation takes place as there's this thing called DNA photolyase, which comes in, which is activated by light and repairs thymine dimers.
Okay. Okay. Good attempt. So so we have got the thymine dimer formed, which is okay. So what will happen because of this? Again, there will be a bulge formation. Okay. Now the person needs to repair this thing, for which DNA photolyse is available. But the thing is that for this enzyme to get activated, again the light is required. Okay. So the energy taken from the light is required by the photolyse to break this bond. Okay. To break the bond, you need light, and that's why in order to get repair, the light energy is required. Okay. अब इसमें ये नहीं समझना है कि वो ट्यूबलाइट से काम चला लेगा. Okay. Now can you justify, Tanvi, if I'm asking you, मैं उसको बहुत अच्छा ट्यूबलाइट का एक्सपोजर दे रही हूँ, still that person is not able to get repair pyrimidine dimer. So what answer will you give to justify? Anyone else? Akshaya, can you try giving the answer?
Ma'am, it's just a guess. Like the DNA basis absorbs light only at 260 nanometer. So the wavelength needed is not provided by tube light to activate the photolyase.
Okay, but here it is the photolyase which is breaking the bond. It's an enzyme.
Yes.
But yeah, it was almost correct because
So protein.
Protein also, see, they give the absorbance at 280 nanometer. It doesn't mean that they will take that energy. But this is also true, like what you said. It is because of the wavelength which is present. So in the sunlight, we have got the wide spectrum, wide range of wavelength is there, whereas tubelight is only giving us a visible wala. Okay, so that is not sufficient. See, higher the wavelength means what? Lesser the energy, if you remember, right?
Yes, ma'am.
So because of this, we need sunlight. So out of which the enzyme can take the lower wavelength wala range so that it will have more energy. Okay, yeah, good. It's good that you guys are trying. Okay. Okay, so there are in the so these are the in the most direct of these systems. A repair enzyme simply reverses or undoes the damage. Okay, so now there are two types of the repair system. One is the already we have talked about nucleotide. Sorry, not discuss. We are going to discuss base excision and nucleotide excision repair mechanism we have, and then we have got the recombinational method. Okay, so in the table as you can see, हमने अभी तक सिर्फ mismatch पढ़ा है, photo reactivation पढ़ा है. Now we are going to discuss about the base nucleotide, then the double strand break repair and the translation. So these four is now what we are going to discuss. Okay. Okay. Then last one is the translation. So we'll come to that. So direct reversal of the DNA damage. So this is first one. So already एक हमने direct reversal में कौन सा आता है? Thymine dimer. This is another type where you have got a methylated guanine is present. Okay. So sorry. So another example of the direct reversal is the removal of the methyl group from the methylated O6 methyl guanine. Okay. So for this, what will happen? We have got the methyl transferase, it will come and they will take the methyl group and now it cannot be used again. So this particular enzyme will not be used again. So kya hoga? It will go for the proteasome degradation. Okay. So this is how the methyl group, which is not required in this over here, is being removed. Now, if you remember, GATC is getting methylated. So I hope everyone knows that which nucleotide is getting methylated in GATC sequence.
C.
Which one?
C, right?
No, it is A. C is getting methylated in your where the gene transcription is taking place. Okay, that is for that to silencing the gene. But when GATC sequence, when you talk about that methylated parental strand and not methylated wala, tab aapka adenine hai jo methylate ho gaya. Okay, so don't get confused. When we talk about CPG island, remember that it is getting methylated because wahan pe we want that area to be silenced. There it is cytosine which is getting methylated. Okay, it's okay. Now let's talk about the base excision repair enzyme. Okay, so now about the base excision repair enzymes and damage is being removed by base flipping. इसमें बहुत simple है. From the word itself, it's very clear that the base which is not required, जो एक mismatch है, it will be flipped out. ठीक है, बाहर की तरफ उसको निकाल देते हैं. So the most prevalent way in which DNA is cleansed of damaged bases is by removing and replacing the altered bases. Okay, so there are two mechanisms, either the base or the nucleotide. So right now we are going to discuss about the base excision repair. Now in this important enzyme is your glycosylase. Okay, glycosylase. Remember UDP glycosylase? एक बार एक क्वेश्चन पूछा था. So it is present in your DNA repair but not in the replication. We have done one question like that. So the enzyme called glycosylases recognizes and removes the damaged base by hydrolyzing the glycosidic bond. So what it is doing actually, so you know that you have got the base, you have got the ribose sugar and you have got the phosphate. So ये क्या करेगा? Glycosidic bond it is breaking. So over here it is breaking and removing. Okay. So a resulting A basic sugar is removed from the DNA backbone in a further nucleolytic step. So what is happening? At the end, you just have got the sugar and the phosphate group. हम क्या करेंगे next? हम इस चीज को ही पूरा निकाल देंगे. ठीक है? Later again new nucleotide will be added against the template and it will be resealed. तो ये हो गया आपका basic कैसे your base excision repair mechanism is working. Okay. Now let's see in detail. Okay, look at the figure over here. Just try to understand. आपको ये structure याद करने की जरूरत नहीं है. So this is an example of uracil glycosylase reaction. Okay. We'll go slowly so that you don't miss it. So this is your backbone, okay, which is shown. This is your backbone. Okay. Now, can you see that in the DNA backbone, by mistake, uracil is present? Okay. Now, uracil is present. So I'm going to remove this thing. So kaun aayega? Uracil glycosylase is going to come. Okay. So uracil glycosylase is going to remove this base. Now, can you see it over here? There is no base. So this sugar and this phosphate which is present, now this become a basic nucleotide over here. Can you see? So it is either, now since uracil is pyrimidine, so you will say that it is a pyrimidic side. If it was purine, you would have said a purinic side. Guys, are you with me? Able to follow?
Yes, ma'am.
Okay, just stop in between, okay, if you're not able to follow. So, so now you can see this site has become now a pyrimidic site. Okay. Now, next, your AP endonuclease is coming. It is making a cut at the DNA backbone. Can you see? At the 5' position. Okay. Now, what will happen? It is going to have, so what is happening? So, AP over here, endo and exo. Finally, 5' end is free over here. So, यहाँ पे क्या होगा आपका 3' free हो गया because यहाँ से phosphate हमने cut कर दिया है. Okay, so here your 3' group is free and here you have got the 5' phosphate group free. Can you see? The entire sugar and phosphate is being removed. Okay. Later, the gap is filled by the DNA polymerase 1 and ligase will come and reseal it. Is it clear, guys?
Yes.
Okay. Since you guys are not asking question, now I'm asking the question. And the question is, why the system has got 3-step mechanism? Okay. पहले वो ग्लाइकोसिलेज को बुलाता है, फिर वो एपी एंडोन्यूक्लियस को बुलाता है एंड एक्सोन्यूक्लियस को बुलाता है. तो पहले वो बेस हटाएगा, उसके बाद यहाँ कट करेगा, उसके बाद यहाँ करेगा. इससे अच्छा क्या है कि the system would have made a enzyme in such a way that the entire nucleotide will be removed at the same time. ऐसा कुछ क्यों नहीं किया सिस्टम ने? क्वेश्चन क्लियर है? Guys, question is clear at least?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, good. So silence means you guys are thinking. Yes. So what will be the answer?
Is it accessibility? Because it'll require 3 different active sites with 3 different properties has to get in there to be able to do all 3 of these simultaneously.
Yes, very good. This is partly correct, but yes, that is a correct path like you were talking about. So isse kya hoga? Then the system has to make 4 types of glycosylase. Okay. Jahan pe it has to break the entire ये पूरा nucleotide 4 types करना पड़ेगा. अभी क्या है आपका 2 में काम हो जा रहा है ना? So you have got the uracil. पहले आपको uracil हटाने के लिए, see if you suppose I had यहाँ पे कुछ और for example the other bases are there. So obviously each enzyme will need that much big or broad active site so that it can accommodate the entire nucleotide, which is not possible. And you know, active site is hardly a small fraction of the entire protein, correct? So it can only accommodate a small portion so that it can just break the bond or reseal the bond. So having removed entire nucleotide will take much more space and much more energy. So it is better that I do at a smaller level. Plus AP endo and exo. it need not to be specific to the guanine, cytosine or thymine or कोई भी. It can be just AP, endo and exo, right? It can be apurinic and apyrimidinic, endo and exo, बस. So that it can make a cut over here. अगर नेचर को ऐसा सोचना होता है कि मुझे यूरासिल के लिए थाइमिन का अलग अलग करना पड़ता है, then they have to make everything 4 times. So that becomes more costly as compared to this. Correct? Now got it? Or
Yes,
got
ma'am.
confused? Huh? You got the answer, right? So जब भी कुछ like if anywhere anyone is asking such questions like why not this and why not that, you can talk about the specificity of the enzyme and plus always talk about how much it is economic to the cell. ठीक है? अगर कोई काम मैं 2 रुपए देके कर सकती हूँ तो क्यों मैं स्पेसिफिसिटी बढ़ाने के चक्कर में 4 और 8 लगाऊं जब मेरा काम 2 में हो सकता है. ओके, so think in that way. Always the cell is gonna make a economic choice. वो कभी भी extra energy spend नहीं करेंगे in synthesizing protein or keeping a gene separate for it. Okay, good. So I hope everyone was thinking about it and now understood. So these are the approaches you should have whenever you study any topic in bio. Okay, good. Okay, so endonucleotidic cleavage also removes the apurinic and apyrimidic sugar, which arises by the spontaneous hydrolysis. See, this is also the answer now. Okay, so if I had only specific for the base excision repair, then in future अगर spontaneous hydrolysis के वजह से apurinic और apyrimidic site होता तो मुझे उसके लिए अलग से enzyme रखने पड़ते. Now I can use the same endonucleases, right? And they can remove these AP sites. Now, after the damaged nucleotide has been removed, polymerase and the ligase will come and restore the template. Okay. Now, DNA glycosylases are lesion specific and cells have multiple DNA glycosylases with different specificities. Okay. Now specific glycosylase recognizes uracil, इसलिए हमने अभी uracil के बारे में किया. Okay. And another one is responsible for oxo-G. Do you remember oxo-G? Today only we have discussed about the oxo-G. Oxo-G क्या था? Of course, it is a consequence of oxidation of guanine. Remember oxidation of guanine, what will happen then? And oxo-G, okay. पूरा नाम याद नहीं आ रहा. Then do you remember it can base pair with what? So it should base pair with C, but instead of that, it will base pair with dash. भूल गए?
Adenine and cytosine.
है?
Adenine and cytosine.
A थोड़ी ना आएगा. It's a G, right? So G is always it is always base pairing with C only. But instead of that, instead of C, it is going to base pair with T. Okay. जिसके वजह से GC के जगह you are going to have T. GC, instead of that, you are going to have AT का transition. Okay. Please remember, that's why I told you ना, make a chart of it, like oxo-G हो गया या deamination में कौन सा किसके साथ वो base pair करेगा, that is important. Okay. Okay, so cleansing the genome of damaged bases is formidable problem because each base is buried in the DNA helix. So ये कैसे पता करता है? इसके लिए the answer is flipping. So it is going to actually flip the bases outside. Okay. See, can you see in the figure the base is being flipped out? This is a crystal structure. Now this is the base which is flipped out. Okay, can you see it? This yellow color thing is the cytosine. So instead of having over here, वो flip out हो जाता है. Now because of this will show a distortion in the structure and that will be recognized by the enzyme in order to repair. Is this much clear? And that's why it is also known as base flipping mechanism also. Okay. Now the question is coming. What if a damaged base is not removed by base excision before DNA replication? What does it mean then? So in the case of OXO-G, which has the tendency to mispair with a fail-safe system exists. What does it that fail-safe system? It's a dedicated glycosylase recognizes OXO-GA base pairs. Oh, sorry, you were correct. It was adenine only. The transition will come to AT later. Yes, sorry. Okay, so a dedicated glycosylase recognizes OXO-GA base pair, which is generated by the misincorporation of the A opposite to G. Now, in this case, the glycosylase removes the A. Thus, the repair enzyme recognizes A opposite to G as a mutation and removes the undamaged but the incorrect base. Yes, are you able to understand what is happening? Just see the figure. अगर OXO-GA repair हो रहा है, ठीक है? कैसे होगा? So oxidation had happened. Okay, it was unexpected. OXO-G यहाँ पे बन गया. Now what will happen in the next round of, first of all, OXO-G will go for the base excision repair. अगर नहीं होता है, okay, the answer is, the question what we were asking was, see, if suppose this is the, it should undergo a base excision repair because the base has been modified. This is not the required one. So it should undergo base excision repair by flipping the base, and then it should be removed, okay, with the help of the glycosylase, etc., etc. If in case this is not working, okay, this first scenario, then the second scenario, it should take place. So what will happen in the replication here? You are going to have GC normal. This is normal. Here the G oxo is going to replicate. It is going to base pair with the A. Okay. Now this base pairing will be recognized by the fail-safe glycosylase. This is the name of the enzyme, and this is going to remove A and insert G back, still keeping the G oxo, which will be later again repaired with the help of the base excision method. Clear? Is it clear? और confused? मैं फिर से एक बार बोलती हूँ, ठीक है? Okay. See, normally अगर जब आपका DNA replication हो रहा है, because of the oxidation, यहाँ पे आपका base G oxo बन गया, ठीक है? It should undergo base excision repair mechanism to become normal. In case this is not happening, okay, then what will happen in the replication? You are going to have one strand normal, and the another one is going to have base pairing with the A, G oxo with A. Now this distortion, okay, this distortion base pair will be recognized by the another enzyme called fail-safe glycosylase. This fail-safe glycosylase will remove the A and have C inserted. Okay, at least the sequence is intact. Correct? Then again, this will go back to, it will go back to base excision repair. So your G oxo will be removed then. Now it is clear?
Yes.
हाँ, point समझ में आया? So ऐसा नहीं है कि अगर एक आपका mechanism fail हो गया है, so the mutation will be accepted. There are many more are there, okay, in order to check. एक checkpoint के लिए दूसरा है, दूसरे के लिए तीसरा है. Eventually जब सारे fail हो जाएंगे, then only the mutation will be accepted in the cell, in the DNA. Okay, yeah. So same way we have got for the other also, like where another example of fail-safe system is that glycosylase which removes T opposite to G. ठीक है, the same way है. Now, such a TG mismatch can arise as in the case of spontaneous deamination, 5-methyl cytosine can become T, right? But T and G तो normal basis है, तो उसको कैसे समझेगा? So whenever there is this TG का mismatch, then only this glycosylase system will come and it is going to replace T with C. Okay, so the enzyme understands that TG का base pairing गलत है. So this they are going to understand and T के हटा के C रख देगा. So this is again done by the fail-safe glycosylase. Okay, yeah, is this much clear to you everyone, guys? Clear? कोई doubt? So we have discussed about the photoreactivation, that is the direct reversal of the mutation. We have also talked about the mismatch repair system and the base excision system. Okay. So till here, I hope everything is clear.
Yes, ma'am.
Okay.